Hello, and welcome to Module 3.3, Real-Time Phylogenetics with UShER. This presentation is a part of the COVID-19 Genomic Epidemiology Toolkit from CDC's Office of Advanced Molecular Detection. My name is Dr. Russ Corbett-Dettig, and I'm a professor of biomolecular engineering at the University of California, Santa Cruz. This module is a part of a collection of training materials and resources meant to help you begin analyzing SARS-CoV-2 genome sequence data. Be sure to check out the Toolkit's other modules as well. In Module 1.3, we covered the basics of building and interpreting phylogenetic trees, and we briefly introduced NextStrain as a powerful tool for interactive visualization of phylogenetic trees. As viral genome sequences are rapidly accumulating, such phylogenetic analyses are increasingly fundamental for interpreting viral transmission dynamics. However, the volume of data has made phylogenetics increasingly challenging. In this module, I'll describe a tool that my team has developed to facilitate real-time phylogenetics of newly sequenced genomes. It's called UShER, for ultra-fast sample placement on existing trees. Briefly, UShER is designed to do three things. First, UShER accurately places samples onto a global phylogeny. Second, UShER constructs new subtrees when added samples are closely related. And third, UShER enables easy visualization using a web interface. Importantly, UShER is fast enough to place sequences in real time. Samples are typically placed in just tenths of a second, and as a result, our tool can empower rapid genomic epidemiology. Before I get into how UShER works and why it's so fast, let me walk you through why a pandemic is challenging for a typical de novo phylogenetics workflow. For a simple illustration of how phylogenetic trees are inferred from molecular sequence data and a description of their parts, be sure to visit Module 1.3 of this toolkit. The way this typically works is that a user accumulates their dataset, then infers a phylogeny. But by the time your tree is ready, in a pandemic, it's already out of date, and a whole pile of data has to be added to it to interpret newly sequenced genomes. So you can add these new sequences to the dataset and start over to infer a new phylogenetic tree from scratch. But the same thing happens again, and your phylogeny is already out of date by the time it's finished. Moreover, with each addition to the dataset, de novo tree inference becomes harder and it takes longer. UShER solves these problems by using a new approach for phylogenetics, where we add newly sequenced samples to an existing phylogenetic tree, rather than inferring a complete tree de novo. To start, UShER takes as input a phylogenetic tree and a set of samples. In this example, samples are labeled A through D, with each sample represented as a list of its mutations relative to the ancestral genome sequence, which we can get from a multiple sequence alignment, as is used by most phylogenetics applications. So for example, at the second position in this alignment, sample D carries the ancestral sequence, and all others have a substitution to U at that site, represented by the phrase to-U in the list of mutations at the right. UShER uses this input to infer the positions of each mutation on the input phylogenetic tree. For example, this mutation circled in red is the one that occurred at position 2. The substitution to U is shared by samples A, B, and C, so we can surmise this mutation occurred on the branch of the tree where these three samples are descendants. We call this finalized object a mutation annotated tree, where each branch is labeled with a number on the tree and the mutations that UShER inferred to occur on that branch are listed to the right. Then, when a new sample is sequenced, rather than infer the full tree, UShER is able to add this new sample to our existing tree. To do that, we provide UShER with a list of mutations present in the sample. UShER compares that sample to every branch in the tree and identifies the branch that requires the fewest additional mutations. In phylogenetics terms, we call this the maximum parsimony placement. Here, you can see that this sample shares one mutation with sample D, so it should be placed on that branch. Importantly, UShER is then able to write the new phylogenetic tree, including the user-supplied sample. And of course, all large phylogenetic trees almost certainly contain errors. but we have found that our approach has comparable accuracy to more conventional de novo phylogenetics methods. You can read more about this in our manuscript, for which I'll provide a link at the end of this presentation. We have found in general that sample placements onto the tree are exactly correct in 97% of cases, and when they are wrong, the placements tend to be very nearby on the tree. So we expect that most users will want to closely examine the placements of newly added samples. UshER therefore outputs a subtree of the 50 samples already in the tree that are the most closely related to the user's samples. Here I'm showing you what those trees can look like using the auspice interface, where I highlighted the user-added samples in red to distinguish them from other samples in black. In this case, we see that the 3 user-uploaded samples are each other's closest relatives, possibly suggesting transmission among the sampled individuals. More information about using NextStrain and exploring the auspice interface can be found in Module 3.1 of this toolkit. In addition to accurately placing high-quality samples, UshER can be used to quickly identify and remove low-quality sequences. Here I'm showing you a results table that our web interface produced when UshER attempted to place user samples. The first metric I want to draw your attention to is the parsimony score. This is the number of mutations on the branch leading to a user's sample. When the value is high, our output will highlight the box in red as shown. This generally suggests that the sequence has many errors and sample placements might not be reliable. In the toy example here, you can see that makes it so that sample E is added to the tree on a very long branch with many unique mutations. The second quality control statistic is the number of equally parsimonious placements for a sample. That's the number of placements on the tree that require the minimum number of added mutations. You can think of this as a measure of the uncertainty inherent to a sample. Usually this happens when a sample has been contaminated with the strain that differs genetically. And in the example shown here, because each site has missing data, placements on each branch are equally good fits for the sample. So to make UshER easy to use, we provide a web application that takes a sequence upload from a user, places samples onto the tree, and then displays a summary of the results as well as subtrees for each sample or set of samples provided by the user. To upload your data, simply select the Choose File button that I've highlighted here, and choose your FASTA file containing up to 100 SARS-CoV-2 consensus genome sequences, and then select Upload. Note that you are uploading samples to our interface, so the sequence data do leave the computer you are working on. We do not share your data with anyone, and we delete the results unless you choose to save them. If you want to have your samples permanently incorporated into the tree, we encourage you to upload your sequence data to the public sequence repositories such as NCBI and GISAID, from which we draw the data that we use to maintain our comprehensive phylogenetic tree. To show you how this works in practice, I'm going to switch over to another tab on my web browser and show you a demo of UshER in action. I've opened a new tab on my browser. And to get to the UshER web portal, I suggest you just type UCSC UshER. And the very first link that pops up will be our web portal. You can also get here using the link I'll provide at the end of this presentation and the link on the web page associated with this module. Now to upload your data, select the Choose File button and navigate to the place on your computer that contains all of your consensus sequences in FASTA format and select that file. You'll notice that UshER has a drop-down menu with different public trees that you can add your sequences to. We do this so that we'll maintain older trees so that you can reproduce results from previous analysis. In this case, I recommend you just use the top most up-to-date tree and select the Upload button here. And UshER will take just a few seconds to add your samples to our global tree and compute summary statistics about those samples for you. All right, it's already done. So we've uploaded 5 samples, and each of them is shown in this summary table here with various statistics computed for that sample. You'll see that each of these boxes is green and one is yellow, suggesting these are reasonably high quality samples. So for example, this column here is showing you the number of bases aligned to the reference sequence. And if you hover over the little question mark, you'll see a description pop up of what this statistic is telling you about this sample's alignment and placement on the tree. If we go over here, you'll see the 2 statistics I identified earlier as particularly important. They're the number of maximally parsimonious placements on the tree and the parsimony score associated with the sample, or the number of mutations on the branch added by adding that sample. You see, both of these are relatively low numbers. There's exactly one equally parsimonious placement for each of these samples, and they add relatively few mutations to the tree. So these are pretty high quality samples. If we scroll down, UShER will produce results for you for each set of related samples here. And so it has a number of useful statistics. For example, the path of the mutations from the root of the tree to your sample, and also the most closely related sample in the public tree and the pangolin lineage that's been assigned to the sample in the public tree. And if you want to view this result in auspice, select the View Subtree 1 in Next Strain button here. And you'll see that our tree pops up in the Next Strain auspice interface. By default, we color every sample in the public tree using the pangolin lineage. So it's kind of a rainbow of colors. If you want to make your samples pop out against this background, I suggest you select the color by and sample type option here. And what will happen is that the user uploaded samples will be highlighted in red, and every sample already in the public tree will be in black. So in this case, you can see that our user added samples are 3 that are very closely related to one another. In fact, 2 of them are genetically identical, here and here. And the other sample is very closely related here, suggesting these may constitute a recent set of transmissions within a local community. You can also see that the most closely related sample to these 2 is here. It's a sample from Minnesota, and information about it pops up by hovering over that sample as well. If you want to see where your other samples place in the global phylogeny, if you scroll down, you'll see that the other 2 samples that we uploaded, remember there were 5 total, are placed in another subtree, subtree 2. And we'll compute the exact same statistics for that set of samples as well. And if you select the View Subtree 2 in Next Strain option here, a new tab pops up again, showing our subtree that we produced in the Next Strain auspice interface. And if we do the same sort of thing where we go to the Color By menu and scroll down to Sample Type, you'll see our samples highlighted here in red, and every sample already in the public tree in black. And in this case, you see that the 2 samples that were uploaded by the user are pretty closely related to each other. And they're also very closely related to another sample from Florida that you can see information about by hovering over here. So to quickly recap and summarize, UShER is a tool for rapidly placing new samples onto an existing phylogenetic tree. The main use we foresee for public health is quickly determining relationships among samples and identifying close relatives in the existing phylogeny. UShER can also be a powerful quality control tool. The URL to the UShER web portal is listed again here and on the Toolkit webpage. There, you will also find links to example data for hands-on exploration of UShER. So UShER has really been a team effort, and a lot of people deserve credit for this work. In particular, I want to thank Yatish Tarakia, who wrote and maintains the core UShER program, and Angie Heinrichs, who developed the web interface. I also want to thank the rest of our team and various funding sources that have supported our work. This concludes Module 3.3. Part 3 of the toolkit focuses on useful tools and skills for applying SARS-CoV-2 genomic data in epidemiological investigations. Please visit the COVID-19 Genomic Epidemiology Toolkit page, where you can find further reading on the topic. On the Toolkit page, you can also subscribe to our mailing list and receive announcements as new modules and materials are released. Thank you.